So let's get started. Thank you for your patience. I know this building can be hard to find, but I'm Kristen Field, and I'm with the Nanobio Interface Center. We run the Genomics for Teachers series, so you'll have a handout on our May session, and we do this during the academic year about once a month. And this is a special session that we're running with the Philadelphia Science Festival. And I'm not sure how you found out about it, but we're really pleased you're here. Let's see, if you've all signed in, so that's good. And I think on our website, we actually, as you see, we're videotaping this on our website. We have videos from previous sessions, so we hope you check that out. We hope you refer that site to your colleagues. And today, I think we've got a really exciting session. We normally only do these for an hour, so this is a hands-on, a little bit extra, activity-based session. And Dr. Junhyong Kim in the Department of Biology will be leading it, and along with his students, Sarah Middleton and Jean Rosario. So welcome and thank you.
Great, thank you very much, and thanks for coming. I guess as Kristen just said, that what we want to do today is I'm happy to talk to you about genomics, which is the research that I do, and any aspects of genomics you might be curious about. But what we've been working on for the past few years is actually trying to develop some teaching models for genomics in the high school classroom. We work with the Franklin Institute on various aspects of this as well. And especially in my lab, what we do is bioinformatics, that is using computers to look at genomic data. And some of that, I think, is more accessible to the classroom because it's not necessarily based on wet lab work where the equipment might be hard to get and so on. So what we wanted to do is to try to give you some exercises that we've developed which may or may not be very useful to you in the classroom. And what I'll do is I'll just start out with about 10-minute introductory talk on genomics as a whole and why we do what we do. And then John will talk about some of the web-based resources that you can access and links and so on that might be useful for you to talk with your students in the classroom. I'll go back to then talking a little bit about bioinformatics and how we use computer programs and try some program. It's not actually programming, but some of the problem-solving exercises that we always do in bioinformatics. And then Sarah's going to talk about some of programming resources. I know, of course, getting students at high school stage to do things like programming requires a lot of motivations on their part and so on. But there are, again, a lot of web-based resources if you can get your students pointed towards the right way that they might be interested in following up on these things. So, actually, I should start with introductions first. I'm sorry, I went off. Can we start over there and just
My name is Danielle, and I have come from the field of science and technology, and I am pursuing and doing a long-term position for a middle school science teacher to also fill out and global need for STEM students and tutoring. So I'm currently trying to see test it out, but also interested in STEM and inspiring children for careers and stuff.
Great, excellent.
My career path.
My name is Catherine Klug. I teach biology in the school district. Genetics is my favorite unit, so that's what brought me to this.
Great.
Hi, I'm John Boyle. I teach chemistry and physical science at Billy Joseph Marie High School for Girls in Holland, PA. And actually, I heard Catherine sent me a note about it.
Cool, excellent. Well, as I said, I'm Junhyong Kim, and I'm a genomicist and computational biologist.
Jean Rosario, I'm a second-year graduate student. I work with dendra localization.
I'm Sarah Middleton. I'm a fourth-year grad student in the Genomes and computational biology program here, and I'm studying RNA biology of neurons and also localization to dendrites and neurons.
Great. So And everything we're going to talk about today, actually, we brought some files and put everything on the USB so you can take the PowerPoints and any of the exercises files that we made, so you may be able to use it in the classroom. So I'll just start out with some introductory slides. That's of course, I think we all know that the genome is a string of DNA, and the DNA is represented by the letters A, C, G, T, the nucleotides. And the important thing, of course, is that like English letter, the particular sequence of the letters matter for the information content of the genome, right? So just as BAT is different from TAB in the English language, ACT is different from TCA, and this is why we want to know the sequence of the DNA, because it's really decoding the sequence that tells us a lot about what information is contained in the DNA and the genome. So the human genome is about 3 billion DNA letters, and if you actually took the whole genome and stretched it out fully, it's about 6 feet long. And although it's all wound up tight in each one of our cells. So I like to tell the students that if you take all the DNA in your body and put it end to end, it will actually go to the moon, back to Earth 50 times. So that's how much string is in your genome, in DNA. And another thing, actually, although I don't have it here, I've done several STEM sessions with high school students, and one of the things that I find very useful is getting students to think about all the different scales of measurements and how small things really are at the molecular level. And I found that to be a very informative sessions with the students, because they don't really realize, say, how small DNA is inside your body. So if you took like a letter of DNA and blew it up into something we can actually touch and size it, like let's say size of a grain of rice, and then you ask how big is your hand compared to a letter of DNA? Well, your hand would be, say, size of the continent of US, right? So that's sort of the size difference. And the reason I make that point is because everything we do in the lab, we're trying to manipulate individual DNA, right? So it's sort of like taking a hand that's the size of the continent of US and trying to pick up individual rice grains, and that's sort of the scales that we work at. So the human body has about 100 trillion cells, and there's 3 billion DNA letters, so nearly every cell has a copy of the genome. So there's 321 zeros, so this is 370 million letters of DNA in your body. And apparently, this is according to the astronomers, this is the same number of stars that are in the visible universe. So you could say that there's as much information in our bodies as there is the universe of stars out there. And of course, as you know, these letters and how these letters work in individual cells in the body tells us how the organism is built up and why each individual cells are different from each other and all the parts of the living system. So in the old days, of course, and actually still is the case, what we really want to know is for all the traits of interest, like my hair color, eye color, or why I might have propensity to have diabetes or heart disease, but we want to know what are the genomic factors that govern these kind of biological processes, right? So we used to start at the top where there's some phenotype that is external measurements of interest, like, say, heart disease. And we want to know what is the genetic information that's related to that heart disease. Right? And so you took a long time into trying to figure out by many genetic crosses and things like crossing the organisms, say, with heart disease, without heart disease, and figuring this out. It took us very, very long time to try to get down to, say, individual genes that might be responsible for that trait. And if the genes involved, if there's hundreds of them, it was very difficult to get down to things like that. But the genomics revolution, which happened from the early 90s on, really gives us a completely different bottom-up view of how to study the organism. So, sometimes we call this the genomic research program because this is sort of opposite of how biologists used to study the organism. So first, we enumerate all the relevant parts in an organism, so starting from the DNA. Right? So sequencing the DNA becomes the first thing to do, and it's sort of the bottom systematic data collection at the very bottom. And then we try to figure out like, things like, well, how many of such things are there? So in this case, usually in genomics, it goes from DNA to the RNA. So we try, and so as you know, this is called gene expression. So in individual cells, we try to figure out how many RNA molecules there are of each of different types. So we make the quantification, and then we try to model how these things all work together by how these different genes might interact with each other, how different quantities of these genes might interact with each other, and eventually go down to associating these parts and all of these into organismal function and dysfunction. So as I said before, previously we started at the bottom and tried to get to the top, but now we do it the other way around, mostly because it's become much, much easier to collect the data from the bottom rather than from over here. So when I was a graduate student, undergraduate and graduate student in the early 80s, we were able to do maybe like, tens of letters, but 1, 2 to 10 letters of DNA sequencing we could do by experiments in the lab per day. And these days, this has gotten to much, much better technologies. So, and most of what we do is involved in the technologies all became taking what we knew how to do actually in the 80s and making all the chemical reactions smaller and automated. So for example, we used to do things like do the reactions and measure the output of the reactions by radioactivity. And that takes a long time because you do something and you put X-ray film on the top to collect the radioactive activity coming off, and it took us like 2 weeks to expose the X-ray film. But now we do it by these fluorescent molecules and shine a laser onto it, and you instantly get a readout of the reaction that has happened. We used to do reactions in standard tubes like this, and then it got to these microtubes, which some of you probably already dealt with, and it became smaller like this. And now all the technologies became even smaller, such that the chemistry is actually run on little etch devices where the technology is the same technology they use for making silicon chips. So all the chemical reactions are done on these kind of silicon chips where things like nucleotides are put onto the chips, and then the reactions are done on the chips and so on. So the newest technologies, which are called, the dominant machine out there is called Illumina sequencing. And the sequencing machines basically has these chemical reactions. Each one of these little dots are chemical reactions that we read off to do in the sequencing reaction. And this is 20 microns, and you can see how many reactions are happening in this dot here. So we now are able to carry out something like about 300 million reactions at the same time in this tiny little space. So we can read 300 billion letters per day compared to tens of letters per day that was in the 80s, right? So in just 30 years, we went from like billion times, actually more than billion times in efficiency of being able to collect this data like this. It's just incredible. I don't think there's anything like this kind of technological, innovation and revolution really. So, you talk about the CPUs having the Moore's law becoming more and more complex. So they double in complexity every 24 months. So every 2 years, the CPUs become more and more complicated. In genomic data and this kind of scale efficiency, that we've been increasing every 18 months, this kind of efficiency. So how do these sequencing reactions actually happen in a sequencer? The main key is, as you know, the base pairing rules. So you have the AT base pairing and CG base pairing. And so if you have a string of DNA at any given position like that G right there that's lighted up, as you know, only one of the bases can form a base pair with that string. So it's a little bit off because of PC Mac transition. But okay, so if you try to attach an A to the G, it won't attach. It will fall off. If you try to attach a T, it won't attach. It will fall off. G, it will fall off. If you have a C, then it will form a bond, actually, to the G, okay? Right? So that's the main thing that happens. And so in a sequencing machine, what we have is, this is the imagine this is the sequence. Now I sort of diagrammatically put these kind of cup and balls to show that how these base pairings can happen. And so we have bases as a sort of pool of chemicals. Each one of them has a fluorescent molecules attached to it, so that we know that the blue ones are A, and say that the red ones are C, and then the green ones are G, and the orange ones are T. And this is the sequence that we're trying to sequence, and of course we don't really know what it is, so we imagine that it's under a black box. And what you do is then you try each letter to see if it fits. And you try each of the 4 letters to see if it fits, and if it fits, then the light, it lights up, so you get this fluorescent light. And so you keep doing this, and then you take a movie of these sequence of lights coming on and off, and then you just play it back, and then you know what the sequence is. And so that's how these sequencing reactions work. And as I said, the main thing is that each of these reactions are happening at nanometer scale and on a glass slide, basically. So each of those spots are where all these lights are coming on, so that we are able to run hundreds of millions or sometimes billions of chemical reactions at the same time in a tiny little glass slide. And that's how we are able to get so much sequencing data at once now. So the first human genome project cost, depending on how you account for these things, something like $2 billion or so. Now we can sequence your genome actually for about 2,500. So that's sort of the efficiency gain. And that's why genomics has become such a big deal, because we can generate the data so cheaply. So the next thing is, of course, since we generate the data so cheaply, we need to know how to interpret this data. And a lot of that involves computation. In fact, all of this data that comes off these machines, you can't even look at it without using computers. So the computers have become completely intimate part of biological research and biomedical practice. Otherwise, we can't do anything. So that's why we want to concentrate on talking about bioinformatics. Okay, John.
So while I'll load this up, I just talk a little bit. So as Dr. Kim alluded to, the amount of data that just produced by the new sequencing techniques and that's been produced lately computationally is just tremendous. And because of this, has been a need to develop tools and resources to deal with this data and to share this data effectively. So locally, people have developed have used tremendous power of the internet to develop tools that can handle this data and can. I'm sorry. Oh, is it not on? Oh, sorry. They can handle this data and people can use it effectively. Luckily, most of this has also been developed for free and can be used by students and anyone really who's interested in using them. And if you give me 1 second, I'll pull up a little document for you. So as I mentioned, these tools. Oh, sure, of course. There it is. Is that good? Can everyone see it? All right. So, so I'll talk to you a little bit about these tools and how can they be used and how can you sort of look at them. So first of all, I guess most convenient place to start one of the most basic tools, and I think one of the most helpful when you're just starting out and you just if you're thinking about using a biological question and we're not quite sure about the details. So NCBI, it has just broad information and a lot of tools that you can use. It can compare, it can align, it can compare a lot of data, either from the genome or for proteins. It can essentially be used for as a basis for you to start a more detailed analysis. You can, if you want, you can search. So you can see you can search for genes, you can search for nucleotide sequences, protein sequences, and you can even do, if I can find it, for species. Really, you name it. It's with biology, you can find it in NCBI, at least a version of it. So it is a really helpful resource and one to always keep in mind. I also like to mention one of the most basic alignment tools, and one that I have used that is also can be found within NCBI, but I think it's fairly important to sort of mention on its own, and that's BLAST. BLAST is fairly easy to use. You just upload a file, their sequence, or you can find one. Let me actually download it. And you can choose and for instance, you can find sequences that are related to whatever sequence you want. And it will take a couple minutes. See how long it takes this time. And it'll give you an alignment. It'll give you, you can do this with either genome sequences, or you can do this with protein sequences. It's very helpful. Another one that I like to, that I've used in the past but I have not used recently is Protopedia, which is a fairly lightweight resource for just basic protein structure. It's very interesting. I do, and I think it's very helpful for as a student, especially when I was starting this, because it allowed me to see. It's something very rare in biology to actually have a visual of what I was studying and what I was looking at. So again, this is not a very, it's not a very sophisticated tool, but it's a very basic tool, and it gives a great visual and a sort of a good way of introducing students to structural biology or looking at proteins in a different way than a sort of this abstract sense of just mentioning a protein or its function. Let's see my marker. Another tool is Ensemble, which is a little bit similar to CBI. It also possesses a lot of data and a lot of curated data. A lot of tools for you to use for analysis, for you to build exercises, or incorporates a lot of new data that. So yeah, I guess I won't spend too much time on that one. Another one that I've, again, not used recently, but I really like, it's the Protein Data Bank, RCBS, Protein Data Bank, which, like Protopedia, it allows you to see 3D structures of a lot of proteins that are but it's more robust and allows you to also compare protein structures if they have them. The only, it's not really a downside, but the only point of this is that it's all curated proteins, which is a good thing, but also means that model proteins that don't have a curated structure are not present here. But there's still, it's still a good way for a student to sort of visualize a structure and to introduce them to proteins in a more concrete way than just something that does biological stuff without any kind of context. And I have another alignment tool that's not BLAST that I actually really enjoy using. And that's called Style, which is very convenient at either aligning multiple sequences, and it gives you a really easy way of just, again, you choose whatever sequence you have, you input them, and it will spit it out, and it will just give you an alignment of what you gave it. Again, this is a very basic tool. It's very convenient. So again, I think this is important. It's important to point out that there's a lot of bioinformatics tools and websites available. It's just impossible for any one person to get experience with all of them. So I'll just give you sort of a bright overview of the ones I personally used. But I think the important thing is you should experiment with them and find out stuff that's convenient to what you want your students to learn and what you feel comfortable using. I guess explore is really the main advice I can give. There's just a lot of resources available. So I have here's some exercises for you guys to do, or for you guys to sort of look at. I guess I wrote this in Google Drive, so I guess the Drive to Windows conversion didn't quite have all the changes correctly, but never mind. I don't know. Should we do the exercises? Yeah, so that'll be fine. So, another resource that I guess I didn't mention earlier was Genontology, which is something I'm personally looking at in my own thesis project. So Genontologies categorize genes by function or some biological property that they share. So it's again convenient if you're sort of interested in a certain function and you're not quite sure what genes might be involved. This is a good way of just starting looking for those genes. So if we go towards this exercise, move forward a little bit. So we'll be looking at the opsin genes, which are the proteins that detect light in your eyes. Specifically, we can look at the cone opsins, the so-called cone opsins, which detect color. So as you guys probably know, you have three opsin genes, which sense red, blue, and green. And so we can look at those and see how they're similar, how they're different using web tools. So I click on Genontology already. Well, before we move on, do you guys have any questions or anything you want to mention or ask? No? Okay. So first of all, I looked for just a geo term or something I'm interested in. Visual perception, I guess, would be the most appropriate for what I was looking for, and just search for it. Now we can go through ontologies, and it will give you a list of the ontologies that most closely match your search terms. So here, we have it right here, visual perception. Pretty good. Now will give me a bunch of lists of genes that are just that have been recorded to be involved with visual perception, whether they're the opsins or not, it doesn't matter. It will give me anything that has been categorized has been related to visual perception. But I'm more interested in the opsins. So I'm going to go here in Panther family, I guess, and I would just click add this as a condition. So I only want to look at the opsins. So I look at the opsin family. So here I can find the first one, the OPN1 medium wave sensitive opsin, which is the green sensitive opsin. So now you have it here. And so now that we have the identities of each opsin, we can actually continue in genontology, but I want to also show you how you can use NCBI to find the sequence of genes. So let's go to NCBI, search for one sensitive. And you can look like this and then click on genes. And it will give me now all the genes of all the opsins from all the species I can find, not just humans. So, all right, let's limit it to humans. So let's just, because the ones I'm interested in looking at. So it gave me the opsins that I want, at least give me some of them. And actually, if I want to look more generally, just look for OPN1, which is the name of all the opsins, all the color opsins in humans. No, give me the wrong one. So, okay, didn't do what I wanted, but technical difficulties. Let's search for them. Well, anyway, I can I can sort of save the ones I've searched to a clipboard and in CBI. That's helpful if you're trying to look for multiple genes. So we'll do that now. Okay, so now, okay, so now I can export these sequences by saving into a file, and ooh, no, didn't do that actually. Search for nucleotide. See it now. Give me the actual sequences.
Make sure it looks pretty. There we go. That's what I wanted. Oop. Your mouse is very sensitive, Sarah. Search. And now I have the 3 protein sequences that I wanted, so I can now save them to a file. So just call them human opsin sequence. You can save them. Okay. Now we can move on to class style, which I prefer to use. It's a little bit easier to use for aligning multiple sequences. And we can go use opsin sequence and submit this. And just to make it a little easier to see, we can add the colors to it. So essentially right now, if you want to sort of generate a hypothesis about potential differences in a set of genes or a set of related genes or similarities in those genes, it's really easy to do. It takes a few minutes, and I think generating a visual like this for the students, it's a lot more significant than sort of just describing something or telling them what you should expect. This is so I detailed how to recreate this exercise in a Word document that you should have in these guys. That's sort of notes about the. There's also alternate approaches that you can do once you identify a gene. As I said, the protein database you can compare sequences might be actually more exciting for a student to do instead of just doing sequences. The structure of the protein might be more visually exciting to see than just see a bunch of letters together. So that's also another possibility to do, but of course still will require genes that would know protein structure, but that would be too hard to do. Another thing that when I was first learning bioinformatics, something that was sort of excited for me to do was build a phylogeny based on a sequence, and that sort of got me really interested and excited about it. So I think the first step of that would be, though, having a comparison. And so you could have a list of very common animals that your students should know what they should be, and you just sort of let them build their own tree based on whatever features they want, and organize how they want, and as long as they have a way of doing it, I think that should be fine. But then, because of all the sequences that are available, you can use sequencing to develop another tree and then just compare them and see how the students fared against a model based on either nucleotide sequence or a protein sequence. In this case, I use 18S rRNA. This is probably not the best thing to use if you want a truly accurate tree, but it's the one I used in my when I first exposed, and I guess sort of a throwback to that, I decided to use it again. So I can show you how to do that. I did this with BLAST just to, again, show you how to use BLAST a little bit and expose you to it. In this case, I already gathered the sequences because it was present in the first exercise. I didn't want you to guys sit through it again. Let's see if they are downloaded though. I can use this. Yep. So from BLAST, just click on nucleotide BLAST. And then align the two or more sequences. Let's just align everything with respect to humans, because we're all humans, so that pique our interest. And then just click BLAST, and the resource will just do everything without you having to guide it or anything. So this will give you an alignment on the 18RS sequences. You can look at it if you want, but usually, again, sort of tedious to look at. It's not really something a student I think might find too exciting. But you can also build a distance tree from it. It'll be more of a proxy of their relationship between these animals, how they are related. So in the end, you produce a figure like this. Again, this is a very simple example, just very few species, just to illustrate what can be done, but obviously much more complicated ones can be created. So here, there's some sort of interesting results that I wouldn't expect. For example, the cow is more related to us than a horse. There is to a horse. So, but again, these are the point of this is not to produce a truly accurate tree of the relationship. It's just sort of for the students to compare and see how different data can give you different conclusions. And really, I think the best way to expand this exercise is just to have to build phylogenies just based on different sequences and different data. Sure.
I'm opening up this USB, and there's two files there, 18S RNA human sequence, which is the same thing you were just speaking of.
Yes.
And one for the mammals, but I can't open it at all.
Oh.
First it converted it to a zip, and then it's non-existent. But I'm guessing these are the codes you're speaking of.
Yeah.
I
So.
can still access them online, though, so it doesn't matter if I can. It's saying I have no file like my Adobe Acrobat.
Okay. Oh, so I'm sorry. So I should have mentioned that. So these are FASTA files, which is a very common file type to save sequences as. You can actually open it with just a text editor, like Notepad or anything. You will have to tell the computer to do it that way, but it should not be a problem opening of them.
So they don't have a program associated with it. It's just a file that contains the sequences.
Right.
You can open it in a text for editing file.
Right.
I tried like 20 ways. now I know.
But any of these web programs will recognize those files and be flirt with it.
Okay.
Yeah.
Shortcut for opening it, you can just change the extent to the file extension to .txt. And then your computer will recognize.
Oh,
right.
It's here on the notepad. It's coming up. Okay. But it would be great for them to see.
Yeah. So sorry, I should have mentioned that.
That's fine.
Yeah. Okay. So again, this is just a bigger part of what you could do. This is just sort of a small time examples of stuff you can build on, and it's but don't feel limited by. I feel like I am not an expert in bioinformatics. I'm just a beginner, but the thing's excited. All right. Thank you. Turn it off.
Somewhat unfamiliar because And there's lots of different things, as John mentioned, that you can explore on the web, but it's not really clear what's going on under the hood. And, you know, what we do a lot is, in fact, we create those programs to do the things like these alignments and things like that. So what I want to do is go over a couple of exercises that I do in my class, where I try to teach the students what it means to program. And first, I try to talk about what it means to program, especially for biology students who never programmed before. And then second, try to work out one exercise at least that has some biological problem that we're trying to solve. So let me just try to do each one of those. So I want to start out with a very classic thing. And the main thing is to add, what I do with my own students is to ask them to try to precisely describe some procedure for something they already know how to do. Right? So for example, here's a very simple example. I have 5 cups. They all have different amounts of water. And I want to rearrange the cups in such a way that they're all in order, like this, right? And so I'm actually going to hand this out to you, to ask you to do this. Okay? Take a few minutes to. Okay. Pardon? Oh, it's what? It isn't like how small number of steps. But that's an interesting question. Maybe. Right. One of those puzzles where you make 3 moves and turn something into a triangle or something like that. I should ask, has anybody programmed before? Oh, great. What do you program in?
Well, I did, I came from, well, I do have SQL Server, SQL language, but C++.
I see. Well, so you know what this exercise is.
Sometimes you want to make the less amount of code,
of course.
The smallest amount
Yes.
to do the same operation.
So, you know, maybe I can just get one of yours. Can I have yours? And what's actually interesting is to have the students switch, and then you ask them, okay, do you understand what the other person tried to tell you? Right? Because the most important part of programming is what we're actually doing is coming up with a very precise language to do something. Right? And that's what really programming is about. They teach you, a very small number of things you can do, words you can use, and sentences basically that you can use. And you use that to actually try to describe how to do very complex things like sort these water cups. So, okay, place the cups together to visualize the levels. Number 2, place the cups from the smallest amount on the far left. Place the cups next to the highest amount next to that cup. Repeat the remaining into the largest. So great. So this is more or less what we normally would do, right? When you except that, of course, now you're cheating because you're using visual computation to say what is the smallest and what is the next smallest and so on, right? So what you're implicitly doing is saying that I can look at this and say, oh, the middle one is the smallest. And then the next one is the second one and so on. In reality, of course, a computer can't do that, right? Computer doesn't have vision that sort of looks at all 5 cups at the same time and does that, right? And so what a computer might be able to do is to take one cup and compare it to the other cup and say which is bigger, right? So in fact, so I used cups instead of numbers because, of course, if I gave you numbers and told you to make it from smallest number to the left to the right, people immediately know what it is because we, of course, know what's the biggest and what's the smallest with numbers, right? Because we practice this over and over again. Whereas with cups, it's a little bit less ambiguous. A little bit more ambiguous as to how to know which is the smallest. But right now, you're sort of saying, well, if I visually look at it, I know what's the smallest. But really, you can't do that you have to say, how do we do that? So one possibility is to say, okay, I take this, pick up the first cup, and look at the second cup and see whether it's bigger than the first or not, right? And then I do the next one, and so on. So if it's bigger, if the next one is bigger, I pick that one up and put this down and keep doing this. And if I do it sequentially, eventually I'll end up with the biggest cup. Right? And then I put that down. And then I repeat this process again. And I'll have to do this over and over again. So you have to be able to describe that. Right? And because that's what a computer does. And so this is called the sorting problem. So you all use Excel spreadsheets to enter students' grade, and then eventually you sort of like go and say sort by name or sort by grade. And that's what the algorithms are doing. It's internally sort of like comparing A's and B's and C's and then finding the last one and then going back and doing this and going back and doing this. And so the main point of this exercise is just think about how to break down the processes in little bits, like comparing this thing, and how you iterate the process over to make a program. And there are many also ways to do it. Another way to do it is these two cups and see which one's bigger. If this one's bigger, we leave it, and we take the next one and compare it. And if the first one is bigger than the second one, we swap them. And then we keep swapping until we get to the end. Then that again should bring the biggest one to the right hand side, right? And then we go to the front and do it again. And we stop right before the last one and do it again. So those are all the different effects. So that's why we call them algorithms. The algorithms are various different way of solving the sorting problem. And there are others. I ask students to like describe how do you add, say, two-digit numbers. And okay, of course, they know how to do this. But actually, if you ask them to describe it very precisely, it actually becomes a hard problem for them to do. All right. So the next one is much more involved exercise. And I'm going to try to describe now in algorithm for doing sequence alignment. And this is what Zhang already talked about, which is the whole idea is that you have two different DNA strings. Like, for example, there we have the ACCA and ACGA. And we want to sort of arrange these two strings in relation to each other, okay, such that we can say one letter corresponds to this letter corresponds to this letter. Okay, so we want to align the two strings to each other. Right? So obviously, in this simple case, it looks like this. We just stack them on top of each other. And by that, what I'm implying that this first position A's and A's correspond, C and C correspond, this C and G corresponds, and A correspond. And if we do that, we see, oh, there's one position difference right there. And you say, oh, there must have been a mutation. Right? So what Zhang showed you was actually there on the web, like taking the three opsin genes, and if you align them, those color things that you saw, is sort of stack the three strings on each other and showed you all the corresponding positions. And from there, you are able to see, oh, those three genes different this place, and that place. And you say, oh, evolution has mutated the genes from each other such that when those changes are made, then one of the genes are able to detect long wavelength, the another is able to detect short wavelength, and another the middle wavelength. Right? So this alignment of two strings is one of the very, very basic algorithms and key algorithms that we do in bioinformatics. Without doing this alignment, we can't do anything. So if I were to take my genome and compare it to your genome, we go and sequence it. As I said, it's only 2,500 to do this now. So then you have to say, okay, what's the differences, right? Obviously, there's going to be some differences, not a whole lot of difference. Right? And so in order to figure that out, I have to take the 3 billion letters from your genome and 3 billion letters from my genome and align them together so I know what the correspondences are. From there, I'll be able to say, oh, that portion's different, this portion's different, right? So this one was kind of trivia. It's not so trivial when it looks like this, when the lengths are different, right? So one way is something, maybe it looks something like this, A, C, and then we introduce what we're going to call gap character, that is it's missing in the bottom string compared to this and this. And so there's two ways to put the missing here, like either the third position is missing or the second position is missing, and they still kind of seem to correspond to each other, right? Now, what you noticed is that when I did this, this is sort of saying, well, what's the best way? I put this in such a way that there's a lot of correspondence between the top string and the bottom string, right? So the whole problem is, how do we find the correspondence? In this case, we can just eyeball it and find the correspondence. Okay? Once it becomes a little bit more involved, it's not so easy. So here's an example. Here's one string and another string. Now it's a little bit longer. It's a little hard to find how to do this problem right now, right? So what we usually want to do with this kind of alignment is that I want to somehow find the correspondence in such a way that string 1 and string 2 is as comparable as possible, right? So somehow they're most aligned well together. If it goes like this, we say they're misaligned, not aligned, right? So we want to align them. Right. So one way to do is we make a table. I'm going to put one string at the top and the other string like this. Okay? And I'm also going to start out with this character, which is called the gap letter character, which means something is missing. Right? So we'll put that there. Okay? We make this kind of table. So remember, this was the string. Yeah? So now the one string's here and one string's there. And then we look at each cell and see, say, for example, this here's a A, here's a C. See if it matches or not. It doesn't match, right? And it doesn't match. A and C doesn't match. A and G doesn't match. A and G doesn't match, and so on. So we look along this table to see if any of these letters match. For here, C matches to this C, right? Okay. So wherever there are matches, we can put a little dot there. Okay. All right. So what this tells me is, okay, these are all the places where this string and this string can match with each other. Right? So here, so again, a block over here. All right. Here's the key step. Okay. So we get the alignment by saying, okay, we're going to figure out how to match this string to this string. And so we're going to take a pass. So I'm going to start here and say, move along any of these. It's like making a chess move, except it's a pawn. The pawn makes only one step at a time. Okay, I can go from here to here, or here to here, or here to here. Okay, one of the three possibilities. And so I'm going to draw a path that goes only from to the right or down. Okay? You can never go back up, or go to the left. Okay, so you can't go like this and zigzag. You're only allowed to go to the right or down. Okay? So for example, I can go down, 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 right, 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 or right, 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 down, 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 or diagonally like this, then down. Okay? I'm going to draw a path from here to here, with the rule that you can only go right or down. Right? Okay. So you get, say, this kind of path, or this kind of path, and so on. Okay? All right. This is the part that's hard. Okay? Each of these paths actually means you can redraw it as an alignment. Okay? All right. I'll show you now on the board. Okay. What this path means is that there's a deletion, and then all of these letters are matched up against the deletion. Okay? And then all of these letters follow after that. So I'll just start at the top. It means C, C, G, G, T. Okay? And all of those things are matched to a deletion in the bottom string. And then it goes all the way down. So then ACCAG. ACCAG. G. So that nothing is matched to anything. Okay, that's what this path is. If you look at this path, okay, this path says, okay, A is matched to this, okay, and then C and C is matched, okay, and then C and C is matched, okay, and then from here we go to A. So again, there's A matched to nothing, right? And then G and G is matched. And then G and G is matched. And then T is not matched to anything again, right? Okay, so that's the alignment. Okay. So what is the algorithm trying to do? It made this table, put all the dots there, and then it tries to find all the paths. There's many different ways of making the path from here to there. It tries to find a path that has as many dots in it as possible. Okay, in this case, I didn't pass through any dots. In this case, I passed through 1, 2, 3, 4 dots. Right? So in this alignment, I had 4 different sets of letters that corresponded to each other. In this one, I had none, right? So this is a better alignment than this. And so what the algorithms try to do is figure out all the possible ways you go through this and find the best path. So I'm going to give you now, again, a table just like that.
So that has nothing to do with complementary base pairing rule. You're just referring
That's right. This has nothing to do with complementary base pairing.
The fact that the program is trying to find the
Matches.
anomalies, I guess. Or
That's right. So we want to find the best matches, or other way is least amount of anomalies, right?
Okay.
So all you have to do is first put the dot in wherever the matches are. So step 1 is to put a dot in that table wherever the matches are. And then you try to find a path through those where you go through the most number of dots. And then if you draw that path, then if you can try to see if you can draw the corresponding alignment, that's also good. But I can tell you that there's over 5,000 possible paths in this table, and that's why, of course, we use a computer to do it. So doing the hand exercise is simply to sort of see what you might be able to do, and of course, you can challenge your students to see who can find the path with the largest number of dots. And you can take the same sequences and put it into the program, the web program that Zhang talked about, the cluster program, which is in the link. And the computer will, of course, automatically find the best alignment for you. So how many did you hit? 10. I don't actually know. I haven't tried it to see. Okay, so, imagine doing that with 3 billion letters now. In fact, our computers can't really do that either. So we have to take lots of shortcuts. Because if you have 3 billion letters by 3 billion letters, that's a humongous table to sort through, right? And even the biggest computers actually can't do that. So we use all kinds of tricks to try to get over the problem. So so bioinformaticians sometimes do things like make these kind of algorithms, and then more importantly, of course, you use the algorithm to then solve biological problems. For example, as I said, we would align our two genomes, see where the differences are. Then we can hope to try to say, well, what are the differences that might have resulted in the phenotypic difference, the trait differences between the two of us, right? Okay, the last thing we wanted to do is that, I don't know about your students, but whenever I dealt with my college students, actually high school students much more than college students, whenever I've done high school students, if you do something fake, like as a demonstration, then they see right through you. They're not interested because it's fake. Right? So what we just did is kind of fake, right? Because that's not what a computer does. A computer doesn't draw lines. You actually have to program this, and then the program actually has to solve this optimality problem, which is to find the shortest, the path with the most number of dots. So you really actually have to learn how to program. So I saw what we would do is that Sarah has been teaching programs to many different people for a long time. So she can describe what resources are available for high school students to get into actual programming. Then they can actually try to program something like this. You can give this to them as a program and say a problem and say, see if you can program this, right? This alignment problem.
So I think most of you said that you have not programmed before. Is that right? You've done a little bit. But so what I want to talk about today are some online resources that your students can use to learn to program, where you yourselves do not actually need to know any programming yourself. So on your USB, there's going to be a handout that will have go through pretty much everything I'm going to talk about now. So the first resource I want to talk about is called Code Academy. And what this is basically an online interactive tutorial that teaches students who don't know anything about programming how to program. And so this website has several different languages that you can choose to learn. Today I'm going to focus on, it's very slow for some reason. Okay, so today I'm going to focus on Python, which is a programming language that is personally the one that I use the most, and it's also the programming language I choose to teach others. And that's because it's actually a very simple and easy to learn programming language. So I want to invite you to follow along with me if you'd like. Does anyone actually want to try this out as I go along, or you just want me to go ahead and do it? Okay. So on the handout, I go through how to go to Code Academy, set up an account. Once you do that, you'll have all these options of languages you can choose, and you just click on Python here. And then you'll see a big Start button, and you just press that. Okay, so I already started it a little bit, so I'll go back to the beginning. And so this is what it looks like. Basically, it provides you with a place to write code, a place where the output of your code will be displayed, and then directions on what you need to do for each exercise. So this very first exercise is just kind of an easy one. It just asks you to hit save and submit. And this is what the students will do every time they have a solution to the given coding problem. So we'll just do that. Okay, so now this is the first real problem. And I already did this, so this is my answer. Okay, so the basic setup for each of these programming problems is that there will be a description of some concept. In this case, it's a variable. It'll give examples of how this concept is used, and then it will provide a set of goals, instructions that the student is to complete using what they just learned about. So in this case, it just asks you to create a variable and to set it equal to the value 10. So I'll just do that. So let's say actually that I made a mistake. Instead of setting this to 10, I set it to 1. If I hit save and submit, this would give me a message telling me, you made a mistake and give you a hint of what you did wrong. And so this is really helpful for students when they're first getting started and they may not realize, what they've done wrong. And likewise, if I were to misspell this, before it gave me a different message. Okay, well, yeah, so it gives you a message that is usually helpful. Once you have it right and you hit submit, it'll say, way to go, and you can go to the next one. And so it goes through a bunch of concepts, and I think it does a really good job guiding students through what they need to know in a logical order, and teaches them, it goes through things that they need to know. So there's several lessons. As you can see, there's about estimated of 13 hours of time that a student would need to complete this entire course. But if they were to do that, at the end they would actually be fairly proficient at basic Python and be able to do a lot of the basic stuff that we do in bioinformatics. And so of course, I don't think that you probably have time to go over this all in your class, but maybe you could have them do a few lessons as an introduction, and then hopefully they get interested on their own. Because the way this website's set up is it tries to make it kind of fun to do what you're doing. You get like achievements for certain, you finish a section, you get an achievement, things like that. And so hopefully the students kind of be self-motivated after they get started. Another great thing about this is it includes several kind of fun, real-life kind of exercises that they can do. So after they finish this first section, they go on to apply what they learned to create a tip calculator. So it makes it a little bit more like, this is what I'm doing has an application in real life. Okay, so any questions about this website? Okay, are your students allowed to create accounts on websites or anything like that in general? Okay. Well, they would have to create an account to use this, so if that's an issue. Okay. So now, this is really good for learning basic programming skills. However, it does not actually specifically get into anything about biology or bioinformatics. And so, to bridge that gap, the second thing I wanted to talk to you about is this website called Rosalind, which gives a series of programming problems of increasing difficulty that are directly related to things that we do in bioinformatics all the time. So this is I think this is a really great website. So the problems are ordered roughly in difficulty, and they build off of one another. So once you've completed one problem, you go on to the next, and it will use concepts from the problem you just completed, and you can kind of reuse your code and keep building. And so I'll just use this first one as an example, counting DNA nucleotides. So this is something that I do frequently. So the kind of format of each problem is that there'll be an introduction to the biology behind the problem, like why we care about answering this question. And then there'll be a more precise definition of the actual programming problem that you're going to solve. So in this case, it talks about a string is a ordered collection of symbols. In this case, it would be a string of A, T, G's, and C's that make up a piece of DNA. And it will tell you the expected input that your program must take, and then the expected output that your program puts out. So in this case, the input will be a string of DNA that is at most 1,000 nucleotides, but of course, since the point is to count the nucleotides, it's not going to tell you how many are there. So you'll have some string of unknown length. And what you must return is simply the number of nucleotides of each type. So the number of A's, the number of C's, the number of G's, and the number of T's. Basically, they click download dataset, and each time they do this, it will be a unique dataset. So if you have your students, all working on this problem at the same time, they can't share answers because they'll all have a different problem. And they download this dataset, they run it through their program, they get the answer, and they submit it, and this website will check the answer to make sure that the answer is correct. So again, you yourselves don't have to actually know the answer. This website will take care of all of that for you. So now, to get back to some of the other things we referenced today, some of the more advanced problems that you can go into here are actually things like sequence alignment. So implementing algorithms that are actually used widely in computational biology to solve this will be some of the more advanced problems that your students can get into if they want. So yeah, so I think that summarizes it. There's a little bit more detail in this handout, but if you have any questions. As a personal note, so I started off as just a biology person, and it wasn't until I got into college that I even got introduced to this idea. I was actually almost more than halfway through college when I got introduced to computational biology, the idea of, the first time I even got exposed to programming at all. And I found myself wishing over and over that someone had just shown it to me. You didn't have to teach me this. Show me that this existed, in high school or something, because I would have just loved it. So I really do think this is an important thing. And being in this field, I see more and more people from other fields, from the wider field of biology, wanting to learn these skills. And so I really think that your students will be well served to just be exposed to these things. So, yeah.
Thank you for everything. Any questions?
Yeah.
I have any questions, but just to say what she said was that, see, I have my degree in biology, and then through my career I ended up with software development and software implementation.
Yeah.
And they don't have a certificate now for computer science, but I'd like to, if I do become, and this is my path, because I'm hoping to teach computer science to middle school.
Okay.
Yeah.
Awesome.
So,
yeah.
To program, but right now they currently don't have. Yeah. And so I hear you saying this normally, it's so true because they actually learn, even in junior or the code project, the code.org, and they're trying to get it into middle school, teaching computer programming.
Yeah.
So then it would pick up into high school. And then they would already have the fundamentals of the language and structures and syntax and things like that. And then they can build upon. And the government is trying to get it into the middle school right now. So
that's awesome.
But it is that problem. That's what kind of inspired me to want to become a teacher, is that I didn't have. You get to college and you're like, okay, I'm interested in this, and my strengths are science and math. But where else? And we don't really do a good job of letting our middle school. We do career day and all that, but we don't do a really good job of letting them know all the careers that could be built to them at a younger age.
Yeah.
So that when they're in college and not trying so hard, I mean, you still could be indecisive and change your path how many times, but maybe just do a better job at reaching them at a younger age, or what careers they can do. It's just not doctors and lawyers. I mean, there's more than out there, and it's not till they get older and they're out in the field and like, oh, I can go this way and that way, and I wish I could have known that. So that's what my career path, I'm not going to go into, but went like this. I went to 1-year medical school even, so that was the first thing, and then I left in 2 years. So like I wish I had known a lot of things that, and when I hear you, I hear a lot of, and that's what's going to happen to them, and if we don't keep trying to. So maybe, and this was seemed more towards high school is what I would say, but I could see relevance that you could pick and piece things to make it apply to middle school.
I mean, they could definitely do it.
Oh yeah, yeah. Because like right now, like why I was interested in teaching high school versus middle school is because genetics was my favorite group also. But now they teach genetics in middle school, so which they start teaching it in middle school. So I was like, well, there's the answer, because I enjoyed working with them and reaching them more because they're more open to be absorbing and at that age, you know. And in high school, I see like you're either in honors or you're like you've already made up your decision if you're gonna what your capability of what your capacity of learning, what you're gonna put into your effort that you're gonna put into learning. But right then, when they're in middle school, they're so open. They're so open to like wanting to know everything, and so I was like, well, this is a shame because I really want to teach genetics too. I want to make sure I teach genetics. So now I was so excited to know that they started teaching them in middle school because I learned it in high school, you know. So and that's where I'm seeing now the whole introducing computer science, genetics, and then there'll be this stuff which you guys, if I don't teach high school, you guys will teach. But I can see that they can still even learn all this, but just not maybe at the scale, you know.
Yeah, even just in like exposure to this possibility is very valuable.
I'm really excited. At first I was, and then now towards the end I can start putting together and going, oh, I can see how I can make this. Yeah. How do you guys feel? Do you feel excited?
Yeah, it's true.
Yeah.
Absolutely.
Yeah. And I'm not even in the path yet. But I just, they're ones that are 6 years, you know, they have more. But that's what encouraged me to want to be. Yeah.
There's also, you know, the WICS, the Women in Computer Science, that local activity that's very, you know, active, and they have visiting days to the computer science department here for high school girls and languages. And there's, I think, also, we've been in our school district trying to start something for, I think, in middle school, girls who code.
Yep, that's what Our Code is, the project that they're trying to do. Very cool. So I don't know if you can get their website still where you're saying the ones that you did study, yeah. So
Great. Yeah. Thank you all for coming today.
Thank you very much.